is very excited about. So yes, we've have the pleasure of introducing Kim Marinelli. Kim, I guess, comes to us officially from her bachelor's, where she did at Easton in Massachusetts at Stonehill College. That was an undergrad in biology, and I guess during that time also had the fortunate experience of doing a study abroad in New Zealand. After that, Kim worked in Sonoma, Santa Barbara County for a few years, learning various aspects of the industry, and I believe motivated her decision to come to UC Davis and join us here for a master's degree. So during that time, Kim joined my lab and Ron Runenbaum's group to begin research as a Plan 1 master's student. She's also done a lot of TAing and engaged in various activities within the larger VEN community. So that includes co-founding the Women in Wine at UC Davis and working within the DEVO organization. And kind of at the forefront of today's talk, of course, is the research that she did during this time. And I wanted to note that, obviously the last year or so, it's been pretty challenging for all of us with COVID and a pandemic, and what that means for all of our lives, both work and personal. In terms of doing a master's research project, this was obviously even that much more challenging. Master's students have a very short amount of time to do their research. And I can tell you, for instance, that in the past year, I think Kim and I have been in the same room maybe once or twice, right? So all of Kim's learning and abilities to, kind of pick up the research has all been through Zoom, which is not a great platform to teach people how to do research and engage with them. Obviously it's been a barrier to learning and picking up, kind of training from other lab personnel due to things like, the 6-foot physical distances that are required, et cetera. But nonetheless, the last 6+ months, Kim has been hugely productive and really has taken control of the project and been completely independent and driven the project to where it is today. So, I have to, kind of applaud her for her, perseverance in the face of this. We've had, research projects canceled this past year because of things. And you'll see in her talk today, what she's been able to accomplish, which I think is establishing a foundation of knowledge and data that we're going to use for years now, kind of going forward to direct further research projects. So thank you, Kim, and the floor is yours to share that research with everyone.
Awesome. Thank you, Ben, for the introduction. Hello, everyone. Kim Marinelli here, second-year master's student. My research has been investigating the effects of oxygenation on redox potential and fermentation kinetics. So I think the motivation for this research can be narrowed down into two main questions. How is oxygen introduction at various times and amounts during fermentation altering redox status? And how is redox status linked to fermentation outcomes? All right, so I'm just going to walk you through an outline or an overview of what I'm going to talk to you guys about today. So I'm going to introduce you to the topic of redox potential and kind of talk about why it's important. Talk about redox as a fermentation parameter and what redox research in the wine literature looks like. I'm going to walk you through my methods and experimental design, the results that have come from this, the conclusions we've made, future research direction, and potential industry benefits that redox has. So just a small crash course on redox potential. So before we get to defining what redox potential is, I'm going to take you back to your general chemistry classes where we talk about what oxidation reduction reactions are. So you may remember the acronyms LEOSIS GER or oil rig, where oxidation is the loss of electrons and reduction is the gain of electrons. So oxidation reduction potential, it's interchangeably called ORP or redox, is a quantifiable measurement of the tendency of a molecule or ion to gain or lose electrons. So ORP is measured and often notated in the literature as EH, and it generally has a millivolt unit associated. So now that we know what redox is, how do we measure it? So ORP is a measurement of electrochemical potential under zero current and represents the status of competing electron transfer reactions. So on the right, you'll see an example of an ORP probe that is today generally used to get these measurements. So this ORP probe, it contains an inert electrode inside, which today is primarily platinum. It's immersed in your solution, and the redox value that's reported is relative to the reference electrode in that probe. So I think it's important to note it that there are different ORP probes, and they do use different reference standards. So two common reference standards are the standard hydrogen electrode and then the silver chloride reference electrode. So your ORP measurements are going to incorporate the millivolt reading plus the offset calculated versus the standard electrode in the probe. I know this seems like a lot of information, but it becomes kind of important and something to be aware of as you look at redox research in the literature, because depending on, these values may vary depending on what kind of probes are being used and if researchers are correcting for this offset value. So a redox reaction that many of us are familiar with is that of the NAD coenzyme, which is the main driver in cellular metabolism. So NAD cycles between an oxidized and reduced states, and it's taking on the role as an electron carrier. So this figure on the right is looking at different species of bacteria as a function of pH and redox. So when we talk about microbiology, we talk about the pH in which different microbes can exist. So you can see here that yes, different microbes exist in different pHs, but they also grow in defined ORP regions. So redox, why is this an important consideration? So given the previous slide, we know redox is important for biology of microbes and that organisms can only live in certain environments in respect to redox. So there's this sort of cyclic relationship between the microbes and their environment. So when we think of the environment influencing which organisms can grow, we also have to note that microbes are influencing the redox around them. So they're essentially manipulating their environment via metabolism. So to put this in context, bring it home in context of grape must. So grape must is going to have a certain redox, which will select for certain microbes that can grow. But when yeast start to grow, they do things like strip oxygen from the environment and produce reductive metabolites and pump out proteins. This can influence the ORP in the environment. So what can ORP tell us, or what can we infer from these redox values? This is a slightly different way of looking at it, but this slide kind of reiterates that there is a close relationship between oxygen and ORP. So because ORP, because of its relationship with oxygen, is intimately connected with what kind of metabolism is going on. So in wine fermentations, we're going to be looking at the right side, where yeast remove oxygen from the environment and they're creating an anaerobic condition. And because of this, we tend to see lower ORP values. So yeast-driven changes in redox, I've kind of put a picture on the side for you here as a visual of all the different reactions that are going on internally in the cell and externally as well. Not all of them, but a nice visual. So things to note is, like I've mentioned, yeast create a reductive environment by depleting oxygen and pumping out reducing compounds such as glutathione. Reductive conditions and fermentations narrow the ecological niche. They're reducing competitions and inhibiting the growth of bacteria in non-Saccharomyces yeast, ultimately allowing Saccharomyces to dominate a fermentation. So an reductive environment can lead to hydrogen sulfide production via metabolism or via spontaneous chemical formation at a low redox potential. So now we've talked about, so based on what I've told you, we can understand why redox might change in a fermentation. So this is a little bit, this is a little window into what redox data looks like. So as winemakers, we're used to looking at these Brix curves where we're tracking fermentation to completion. So this is a depletion of sugar, and conversely, the accumulation of ethanol. We're going to have this red line here, which is our redox curve. We can see as fermentation picks up and yeast metabolism accelerates, we're going to have this drop in redox and a rise when it arrests. So this green curve right here will be representative of your cell density or your biomass. And something to take away from here is that kind of your maximum cell biomass, which we can think of, maximum metabolism, is corresponding to your minimum ORP value. So ORP as a process parameter. What is a process parameter? It's a real-time metric that can be used to make informed decisions about controlling your bioprocess. Process parameters help to optimize and automate your process. And ORP specifically is a mixture or composite measurement that considers pH, dissolved oxygen, temperature, and all other chemical half reactions in a solution. So generally in winemaking today, the process parameters that we can think of when following a fermentation are your Brix, your temperature, sometimes pH. ORP provides additional insight in regards to fermentation health, as well as an insight into biological and chemical status. So in the context of wine, we know there's a lot of important chemistry going on. And as winemakers, these things are things that we have to be mindful of, as they can have impacts on our final product and sensorial qualities. So though chemistry is not the focus of, redox chemistry is not the focus of my particular research, I just want to note that typical wine reactions that can occur in the wine matrix and their relationship to potential. And that these are ultimately redox driven. So redox influence on the transition state of metal, which is iron and copper, and it changes with the complexing of things in the environment such as tartrate. It also determines things like spontaneous formation of hydrogen sulfide from elemental sulfur. So this is a fraction in form plot. So we all know that reactions are going on and they are in part controlled by redox. So I guess this is just another way to visually look at it. Generally in wine, we're falling between that 400 to negative 200 range, where during fermentation, we're falling into the negatives, and after fermentation, we're going back into the positives. And I just think this is a good visual representation of the change between transition states in copper 1 and copper 2, and how that's affected with changes in redox potential. Another interesting thing to note is that I've been talking a lot about your internal redox state of yeast cells. And just to note how far down here it is, it's around negative 290. So I'm going to walk you through or just touch upon some of the interesting research regarding redox in the wine literature. This study looked at the relationship between redox and temperature. They monitored redox. They monitored the redox of fermentations run at different temperatures in both red and white wines. As you can see in both studies, as temperature of fermentations increased, we saw a decrease in redox potential. So this is a couple of studies that really spoke to the relationship between ORP and temperature. So redox and hydrogen sulfide production. So I just want to bring your eyes to this graph over here. So there's a lot of curves going on, but the curves that we're concerned with are this EH or redox curve and this hydrogen sulfide curve. So this is an older study. It was published in the 60s, but it did show that peak hydrogen sulfide production corresponded to minimum SO2 levels. This relationship that is something as winemakers is very important. It's a motivating question, I think, in that if you were able to control this redox level, would you actually produce less H2S? And I think this was a motivation for the study to come. So advanced monitoring and control in wine fermentation. This was actually a study conducted here by David Colleen in 2016 at UC Davis. Guess not here because we're not at campus. It was conducted in the research winery in RTJs, so 100-liter ferment. So this was the first study to demonstrate that redox potential could be controlled, and he did so via aeration. Some of the conclusions that he made were that redox control resulted in faster peak fermentation rates and earlier fermentation completion. So just to point out some of the parts on this graph, we have his redox graph where redox was controlled to a set point and it was able to be maintained throughout fermentation versus that is left uncontrolled. Over here, pointing to our Brix curve, we can see that fermentations finished faster than those that were unaerated. So there was a couple things in this study that Colleen mentioned needed further investigation. He mentioned that now that we're seeing these faster fermentation kinetics, what is the driver behind that? And he proposed that further investigation be done in terms of cell densities and cell viability. He also was interested in what redox control looked like at various set points. So this study kind of laid the ground for my own work. So I'm going to walk you through my methods in experimental design. So this is a picture of the beloved bioreactors at the lab. So my experiments were carried out in these 1-liter bioreactors. I used Chardonnay white juice concentrate diluted to 24 Brix as my medium. I think we did decide to go with concentrate for consistency purposes and that we had it in the winery and I would be able to run multiple experiments across the same medium. Ferments were run at 23 degrees Celsius and the yeast that I used was EC1118. Redox was measured using Hamilton ORP probes and then we controlled redox to a set point. So what does it mean to control to a set point? First, you would pick your desired set point or desired value that you want to hold redox to throughout the fermentation. And then as redox drops below that set point, we pulse in air as needed in order to maintain that number. So I've been talking a lot about using aeration to raise redox. And we did so in Colleen as well. So I just wanted to lay out kind of a little schematic to give you some insight on what's going on and why this is working. So it's just an example, but so the oxidizing potential of molecular oxygen in wine is harnessed by the generation of reactive oxygen species. So the initial transfer of electrons leads to the formation of a superoxide, and at wine pH, that happens to be a hydroperoxide. The activation of this step requires a catalyst, and the activation of this oxygen is presumably due to transition metals, such as iron or copper. The transfer of the second electron would produce hydrogen peroxide. It should be noted here that this redox response, because it is related to metals, will likely differ from wine to wine depending on composition. And so this is our new redox control setup in the lab. And I'll walk you through what we have going on. So we have an air supply, which is a condensed air cylinder. The air from the cylinder travels through the pressure regulator, which is generally set to 40 psi. It travels through a flow meter, which the flow has varied from experiment to experiment. And then from the flow meter, it travels into our box that contains the solenoids. So again, once an ORP probe measures that our redox potential is dropping below that set point, the solenoid will open and release air into the bioreactor in hopes of maintaining that set point. So I'm going to walk you through three different experiments, three of the main experiments that I've run. They vary slightly in experimental parameters and experimental design, so I'll go into the details of them as we get to them. So here are the results. Okay, so the first experiment. So I think it's important to note here that prior to this work, we didn't have the infrastructure or a developed protocol for controlling ORP at the lab scale. So a big part of my work has been focused on troubleshooting and developing that. So we have these new electronics and this new setup in the lab. So my first experiment was done in order to ensure that we actually did have redox control, given the new setup and the concentrate median. My first experiment was run in triplicate, where I had 3 bioreactors that were controlled at a set point and 3 that had no ORP control. So for the first run, we selected a semi-arbitrary set point of -20 millivolts based on what we found in the literature. We weren't sure how much air we would need to pulse in or how long the solenoids would have to be open in order to maintain our desired set point. So at the beginning, we started at lower flow rates, and a shorter amount of time the solenoids were opened. And as fermentation progressed and we weren't seeing the response that we wanted, we increased flow rate and we increased the time that the solenoids were open. All right, so getting to the results, these graphs can look a little busy, so I'm going to kind of walk you through what we'll be looking at for the next experiments to come. So this up here is our temperature line, and that confirming that we've been holding our ferments at 23 degrees. These dotted lines right here are Brix curves, and these solid lines are our redox curves. I think some things to point out here are that these 3 reactors or 3 lines are uncontrolled reactors. And we can see that the redox potential falls to around -200 or a little less. Where those reactors that received air or were under ORP control held at a higher set point. So we have to remember that we were hoping to hold it at a -20 set point. That was not accomplished. That was some great insight. It wasn't accomplished, and but we did still notice this difference between controlled ferments and uncontrolled ferments. And this was particularly obvious too in this division in fermentation kinetics, where these controlled fermentations demonstrated much faster fermentation kinetics than those that were unaerated. So we also did cell counts to kind of speak to cell density or biomass accumulation. So we have our cell counts or these curves, and then we have our solid curves, and then again, our Brix curves are the dotted lines. So in addition to ferments having faster fermentation kinetics, they were also reaching higher cell densities when air was introduced. So if we were to refer back to what I had said about the clean status, where he also saw study where he also saw faster fermentation kinetics, he had also hypothesized that this could be attributed to an increase in cell density. And this data here kind of supports that hypothesis. So from our first experiment, we learned that we could control redox and that we had a faster fermentation. We learned that we could control for faster fermentation and we could reach higher cell densities, but we couldn't hold our set point at that negative 20 millivolts. So using this information in my second experiment, we changed our set point and changed the way we were delivering air in hopes that that would put us somewhere in the range that we would be able to control to our desired set point. So now that we know that we had some aspect of control, we now pose the question, when is adding air important in order to get these fermentation outcomes that we've been seeing? So we did this experiment in 24-hour intervals where we had no air introduction serving as our control for the experiment. And then in 24-hour intervals, we would shut the solenoids off throughout the fermentation. Okay, and again, just to acquaint you, we have our temperature here holding steady at 23. And I guess this curve looks a little busy, so I guess I'm just going to show you all of the data. And I recognize busy, but there are some important points where we learn from. So as we were learning how to set up these fermentations, we did have some technical errors that made our data look a bit noisy. So again, we have the steady temperature, our solid lines down here are redox potential, and then we do have our Brix curves that are dotted lines. So I guess we could start with our 0 hours of control. It's this blue line that we can track and it hits lower redox potentials. You can kind of notice that it tracks with this green line. And this actually made sense to us because in that 24-hour period where the second solenoid would have been shut off, it actually never hit the low enough ORP that it hit the set point and the solenoids would have opened. So really this ended up being a duplicate. And this also tracked as far as they showed very similar fermentation kinetics. And on the other extreme, we can see that the 96 and 120 hour ferments also demonstrated very similar redox curves and fermentation kinetics. So this is because we found that these two were duplicates as well, because at 96 hours, our fermentation was complete. So there was really no difference between the two. I think both of those points were really important because it really helps us to zone in on the time period that we should be focusing on. And it kind of sets the stage for our future experiments. Another really positive aspect of this research was that even though there was those disruptions that kind of knocked their redox curves, we did find that we could hold at the set point of -60. So here It's just another example of cell counts, and that they track as far as increased air delivery, increase the fermentation speeds, and in relation to that, increase the cell densities. Moving on to the third experiment. So we were happy with our parameters that we've decided from the last experiment. And then we had honed in our time frame. So instead of 24-hour increments, we cut it down to 16-hour increments, following it through 80 hours. Something a little different that we did do in this experiment is, in order to ensure that all bioreactors would be included in this data set, we waited for the solenoid to turn on in this bioreactor. And then once it turned on, from then it was a 16-hour control period, and so on. Okay, so this data set ended up yielding some really cool results. It really demonstrated a clear progression of the difference that results from various ORP shutoff times. So again, this is uncontrolled, so our ORP is falling to negative 150-ish. We will note that we were having some electronics issues in the lab, so our temperature kind of varied, but it did hold throughout the rest of the fermentation. So I'm going to show you 16 hours, 32 hours, 48 hours, 64 hours, and 80 hours. So I just clicked through them to kind of show the progression of this nice trend that we were seeing as we shut off air supply. So the great things that came out of this experiment is we were able to really control to that negative 60 set point throughout almost all the reactors. In that these fermentation times correlated with the length of time, or the length of time that they were receiving ORP control. So this data suggests that if you can control ORP in the context of about the first 4 days of fermentation, you ultimately achieve the fastest fermentation rates. We found that controlling even longer, so our 80-hour mark, didn't seem to have much difference in terms of fermentation kinetics, so they did finish at the same time. So we have added another level of data collection in my more recent run. And we started preliminary organic acid analysis. And though it's very, very new, it's a new way of looking at our data and a new protocol. One thing that we thought was very interesting, and a trend that we liked, we were happy to see, was unaerated ferments demonstrated a higher accumulation of acetic acid at the end of fermentation than those that were aerated. So we kind of saw this as the next step, because yes, we were seeing a difference in fermentation kinetics, but ultimately we want to make a connection with some sort of fermentation outcome. So we figured out, if we can figure out what the best strategy is to optimize the parameters that winemakers may be interested in, we think this points to the fact that fermentation time may not be the only parameter that controlling ORP would be affecting. So again, these are our cell count data for this experiment, and this speaks to reproducibility, as we do see a relationship between ORP control, fermentation time, and cell densities. So what can we conclude from these trials or these experiments? Well, in EC111 ferments with ORP control, we're seeing faster fermentation kinetics and earlier finishing times. We're reaching higher cell densities. And the preliminary and granic acid analysis demonstrated greater acetic acid accumulation than those that were unaerated, fermented than it was unaerated. So this is ongoing research, as Ben had said. So there's a lot of different future research directions you could go in. One would be to better define a target ORP value. So, finding the optimal amount of oxygen required to get desired fermentation outcomes. We'd like to look more into wine chemistry in regards to redox control. So what is H2S production looking like, organic acid and amino acid accumulations. We'd like to explore alternate methods of redox control. This could potentially be accomplished by dripping hydrogen peroxide into the solutions. And the next step would be to understand what ORP control looks like under production conditions. So really scaling up these fermentations and using a grape juice median as your, or grape juice as your median. I know wine research is really complete without a sensory aspect. So we'd like to do some sensory evaluations of wine fermented under ORP conditions, under ORP control. So potential industry benefits. So if we're seeing this increase in fermentation rate, could this speak to increase in the rate of tank turnover during harvest? Could it be an indicator of fermentation health, maybe a predictive parameter for stuck fermentations? As a potential to reduce VA character in finished wines. I think this would be particularly important in red wines that we're going to undergo malolactic fermentation, as at the end of malolactic fermentation, we tend to gain acetic acid. We could have reduced formations of hydrogen sulfide production in the presence of elemental sulfur. This would reduce the need for copper fining and/or product loss due to faulty reductive characters in wine. So my acknowledgements and thank yous. A big shout out to Ben Montetit and Ron Renenbaum, my advisors. They have been excellent and have answered all of my questions anytime I have them. My colleagues and collaborators, James Nelson, Gita Malia, Andre Knosen, Bob Coleman, Gordon Walker, and Roger Bolton. The 2021 B&E graduate cohort, they've really made my time here special, and I feel like I have a small viticulture and enology family. The VEN faculty and staff, and some of my funding sources, Wine Spectator, the American Society of Enology and Viticulture, and Jastrow Shields. We can open it up to any questions.
All right. Thank you, Kim. Excellent timing. We have about 10 minutes left for questions. So if anyone out there has a question, feel free to enter it in the chat if you like, and I can ask it, or you can unmute yourself and ask Kim any questions that you may have.
Kim, I believe halfway through you were showing a slide on the Colleen study. I'm not sure if you can go back to that. I think they proposed changes in cell viability based on the oxygen reduction potential.
Oh yeah, he
is that something that you
didn't really
for?
So it's not something I looked for in particular. Well, it was at first, so that was of interest when we did our first I dabbled the first few times with fermentation or with redox control. Though there's probably an aspect definitely there with stressing fermentations. And the fermentations I was conducting, we didn't see a change in viability in the case that redox was falling higher or lower. So we kind of stopped in the proceeding. We kind of focused more on different types of analysis than viability.
Thank you.
Thanks, Dave.
Hey, Kim, great job. Well done. It's been a pleasure to see your research evolve and get to be a part of it. I was curious on the timed redox things when you were doing control. As soon as you cut off control, sometimes you'd see that little bump afterwards, almost as if it was like rising up. Did we ever figure out, was that like a solenoid thing, an air delivery, air pressure thing, or do we have any idea what was going on there?
So that has been the golden question. And he's talking about here, if you can see these little pop-ups towards the end of fermentation where you'd shut off this one and see response, a greater response here. I've been talking to James, who is our, he's our electrical engineer. And so it has to have something to do with our electronics or air delivery setup. What that is, we haven't done another experiment since this one. So I think we'll have to put our heads together in lab and try to figure that one out. But
also, did you do our very high-tech temperature light control strategy by putting cardboard boxes around everything?
yes, and this one I did. So we attempted this experimental setup, this bottom one, a couple times, and though we saw very reproducible results, but in our prior run to the one that I'm showing you now, we saw these almost humps in the redox curves, and they were relating to the weather and if it was raining or sunny, and they were like a cyclic trend. So for our next run, we put boxes all around the bioreactors to make sure that that was not an environmental condition that was influencing our results.
We do the highest tech science here at UC Davis.
I have a quick question. Kim, you'd mentioned some kind of bumps in the data, and I don't know if maybe that's what you and Gordon were just talking about after the solenoids go off, but were there any other kind of like anomalous portions of the reading, and did you attribute those to any other just complications with it being like a living system?
So I think this is the second study is probably a better one. The bumps in the reading was one thing, but this is probably a study where we had a lot of things happen. I mean, this was a new protocol, so we did have events like overflow events. We had environmental events where we were seeing that cyclic kind of pattern due to sun. We did have events where air delivery was shut off or it was too low or too high. So it kind of knocks our, these redox probes are really sensitive. So it would start to knock our redox off from our set point. I think our first two experiments, we experienced that more, our first two or three, two for overflow, three was the sunlight thing. But in that last run that I showed you, we had it looked like a control besides the little bumps that we've yet to come to a conclusion that why that's happening.
Cool, exciting, thank you.
Mm-hmm.
Kim, I know you just did model wine, but I was just curious at the end, did you actually smell like if versus the non-aerated versus the aerated ones, was there much of a difference? I was just curious.
So I think, so concentrate in general has this notion of being pretty stinky. So finished wines, I have to say, I can't talk to the sensory aspect of those finished ferments. I think that you would probably, I'm not sure, I can't speak to it, but you might see a little bit more of a difference if you were actually working with a grape juice medium that wasn't producing wasn't known to produce such unfavorable smells or undesirable smells anyway. I think that ideally we would have loved to have a sensory component to some of this research. And I helped with a little bit of the ORP work that was being done in the winery, but it just wasn't possible this year with COVID.
I mean, you did a significant amount of work in the winery too. You had a bunch of iterations in large tanks and stuff like that, and some of them were a little. We didn't totally understand the set point stuff, but I think it was really valuable and interesting to see the work that you did there too.
Yeah, I helped James and Roger and Andre with doing a little bit of winemaking stuff as they were developing some of their electronics. And I don't have the data for that in this PowerPoint, but we were seeing some interesting trends where aeration we're definitely seeing that fermentation kinetic acceleration aspect, but we were having a hard time controlling at a set point, and compositional questions about the wine's composition and presence of transition metals and their ability to react to oxygen did come up. So we decided after harvest was done that we would move to concentrate and kind of focus in on the lab scale so we could get some control.
This was awesome, Kim. Thank you.
Yeah, thank you for listening.
All right. Any other questions floating around out there?
I'll jump in, Ben.
Sure, Bob, go ahead.
What's the future hold for you? What are your plans?
For next experiment?
Well, this in your winemaking career.
So, I mean, I'm hoping before I graduate, we'll get a couple more runs in and we can kind of explore that set point question a little bit more. And then I got to get to finish that thesis up. And I am, and then I'll be going on to work at Opus One in the end of the summer. And I'll continue as a research intern working on redox potential stuff in the winery. Be cool. It'd be a little bit more of applied science.
Awesome job, Kim.
Thank you.
All right. Well, if there's no more questions, thanks, Kim, for a talk. Virtual hands. You can unmute yourself and cheer whatever you would like.
You. There you go, Kim.
Thanks, guys.
And thank you all for
Thanks
being
for
here.
listening.
Well done, Kim. Very nice.
Thank you.